Hi guys, in this video I'll be talking about the replication, basically the translation of the HCV single-stranded RNA genome. What happens? First of all, this is the single-stranded RNA genome, okay, that has only a single ORF, a single open reading frame, okay. Now they are flanked by two regions, one that is 5' NTR, one that is 3' NTR. Now NTR stands for non-translational regions, okay. Apart from the 5' NTR, at the close proximity of the 5' NTR, there is another stem loop structures. Sorry to say, but I'm a very bad painter. What should I say? Okay, please don't mind. So this structure, this stem loop structure is basically IRES, means internal ribosomal entry site. Okay. So with the help of this IRES, HCV genome, they will start their translation with the help of host translational machineries, means host ribosome that will be recruited to this site, and with the host proteins they will first translate their own single open reading frame, okay. Now that will give rise to a single polyprotein. Now this is basically a polyprotein. So that polyprotein basically consists of 10 proteins. What are the 10 proteins? I'll again draw this genome, or sorry, it's basically a protein. So 5', 3'. This is basically started with C, the core protein, E1, E2, then P7, then NS2, NS3, a very small region that is NS4A, then NS4B, then NS5A, and last but not the least NS5B. Okay, so this is the total polyprotein. But with this polyprotein, virus cannot do anything, so they need single proteins from this. So automatically it's a common sense that some peptidase, they must cleave here, and here also, so that it can give rise to 10 different proteins from a single polyprotein. Okay, why single polyprotein? Because there is a single ORF. There is no other ORF, not a specific ORF for core protein, not a specific ORF for E1 or specific for E2. No, it's not like that. It's a single open reading frame which will give rise to, after translation, a large amount of single polyprotein, and after the proteolytic cleavage and post-translational modifications, it will give rise to 10 different proteins. So what are the proteins? I'll first write it down. This is a polyprotein, okay. This is a core protein. Core means that will give rise to capsid proteins. E1, I have already told, E1, E2, they are present as the antigen glycoproteins. Okay, so these three, particularly these three, they become structural proteins. Okay, so these are the structural proteins. Now P7, they basically forms porine proteins. NS2, it's basically, they have autoprotease activity or autocatalytic activity, autocleavage activity, okay. Then there is NS3. NS3 is also a protease. NS3 is basically a protease. NS4 is a cofactor. Okay, this NS4, that is a cofactor which will bind to this after post-translational modification. And NS4B, then NS5A, 5B, this all from NS3 to NS5B, they are the non-structural proteins. Now non-structural proteins means they are basically participated into replication of HCV replication, And those proteins which help in their replication, they are obviously non-structural proteins. And structural means those proteins which actually help to make the nucleocapsid, the envelope, the antigens which are present on the virus particles after their release from the host cell. So these are the 10 different proteins after the post-translational modifications after this polyprotein cleavage. Okay, now what happens? Basically, host cell proteases, the signal peptidase, they will cleave this C, E1, E2, P7, etc. Then NS3, they will play. How? Because these are also proteases. NS3, they will cleave at this junction, NS4A, NS4B, between NS4B and NS5A, and between NS5B and NS5A. All these junctions from here, started from here to here, are cleaved off by NS3. And once NS4A, the cofactor of NS3, has been cleaved off, they become free. Okay, whenever they becomes free, they always make a bond with the N-terminal domain of NS3. So that NS3 along with its cofactor, now they will become activated and they will make other cleavages to make this polyprotein much more active, and further they will help in the replication cycle of the HCV inside the host cell. So this is it. If you like this video, please subscribe my channel and share with your friends. Thank you so much.